So we can start slowly, and also I'm gonna first briefly talk about I will give an announcement. So welcome everyone to the community meeting. And I would like to share very briefly my screen. So I would like to announce here, it's kind of the second time we're mentioning it, but it has been happening and we're organizing it. And we're talking about the first ESVERS conference that will take place in Munich on 10th to 12th of September. And I think that people that they are attending the community meetings are the most relevant audience for this conference. Soon we will start advertising it as well on the social media, and very soon the registration will open. We will accept abstracts for small talks and posters. And the idea is that we will dedicate one day for some speakers, keynote speakers or invited speakers anyway, but the next days will be mainly dedicated to the community and the members and the users with workshops and Q&As, panels, and that's kind of the idea for now. And we're still shaping it, but it's getting there. So keep an eye. We're going to post it everywhere, of course, but keep an eye and save the date if you really want to join. Because we have kind of a limited capacity, especially because it's the first one, and we didn't go big, so we're a bit, we will reserve some spots. So yeah, that's the announcement. Save the date, 10th to 12th of September, the first ESVERS conference. I hope I will see you there. And with that, I can move to the next topic, which is the main topic. So Doron, welcome and thank you for accepting the invitation. Today we have Doron, a PhD student from Dana Pierce Lab, and he's going to talk about ENVI or ENVI. He will correct me when he talks about it. I would call it ENVI, to be honest, because it's already a word. Deep learning based method about integrating single cell with spatial transcriptomics. So he will tell us all about it. Remember to put your questions in the chat or raise your hand, and in the end, I can call your name. So we can see your slides. The floor is yours.
Sorry, I just had to unmute myself. All right, perfect. I assume everyone can hear me, and if they can't, they will let me know. Thank you everyone for coming. It really means a lot to talk to this audience. I'm super excited about ESVERS, and I think generally the biggest impediment to applying any algorithm with single cell data or even beyond that is how easy is it to use it. And a community like ESVERS and the stuff that goes into it is really driving forward the idea of like making stuff easy and simple and straightforward to use. And on that note, I'm now going to be talking about ENVI, which is an algorithm we've been working on with the goal of reconstructing spatial context for single cell transcriptomics data. So I want to start with like a little mindful overview of what it is precisely we're trying to capture. And the mantra that I have in the back of my head when I was working on this is that with a change in niche of a cell, we should also expect to see a change in the phenotype of the cell. So the example that I'm showing here is that we have this like greenish bluish cell that is surrounded by all these green cells. And as the environment of the cell changes from these green cells to these orange cells, we also see that the cell itself changes as it reacts to signals from its environment, and it becomes this bluish cyan-like cell. So there is a relationship between what the cell is like and what its environment is. And this is an idea that is constantly being shown again and again in almost every single field in biology. This is an immune cell going deeper into the tumor and reacting to the tumor cells around it. This is a stem cell reacting to its niche and based on signals from it, deciding which lineage it's going to select. And the nice thing that we have today is that we can now model this relationship between the niche of a cell and its phenotype using spatial transcriptomics or imaging or any of these methods throughout. And I argue that either implicitly or explicitly, the goal of every study that uses spatial transcriptomics is to get at this relationship between the environment of a cell and its phenotype or gene expression. And the idea is that, say you could do this modeling really, really well, and you can perfectly articulate the relationship between an environment and gene expression, then let's say you do just single cell RNA sequencing and all you see is the expression of a cell. Maybe if you did this modeling again super well, you'll be able to reconstruct what was the environment pre-disassociation, right? So to do single cell, you lose any sort of spatial context because you had to disassociate the tissue to do the sequencing. But maybe if you have some spatial data to guide you and supervise what you're trying to do, you'll be able to like see the signals in the expression to tell what was there in the environment. I'm not sure exactly how questions go here, but if people have questions, feel free to raise your hands or say something in the chat. I'll try to answer them as I go. But this is the grand idea, right? Like we can maybe see hints of what was the environment in gene expression alone. So again, the idea is that phenotype reflects environment. And there's a relationship and an interplay between these two concepts. Now, to get at phenotype, we have a lot of tools these days. Most of them have already been implemented by SCVerse and methods like it. So you could do visualization with UMAP or T-STEAM, clustering with Leiden or Phenograph to get at discrete signals in your data. You could maybe look for continuous signals with diffusion components and trajectory analysis algorithms. You could find gene programs, do batch correction. You could do all of this on phenotype, i.e. gene expression data. But when you want to move and ask what is going on in the niche or environments of cells, it's not exactly clear what you can do these days and what are the tools available for that. It's definitely not as straightforward as it is dealing with phenotype or gene expression. And the main reason for that is because environments are multicellular objects. So how does it look like when we actually try to do this analysis onto spatial data? And here I'm showing an example of sequence data of mice grass relation. So what you would do is you would take a bunch of cells in your tissue, or all the cells in your tissue, and you would look at their immediate neighborhood to some extent. And what you would do from that immediate neighborhood is you would just take out the gene expression matrices of those neighbors, right? So I have this cell right here. I have like its 5 nearest neighbors, and I just look at their gene expression space. Here I'm just showing 300 out of 350 genes, but you can extrapolate and imagine what that would look like for all genes in this data set. And this you would do for every single cell in your spatial data set, right? And the way I like to think about this now is that this is the rawest, most unbiased form of environment analysis. I simply took all the cells in my data set. I just straight out took the gene expression matrices of their neighbors, and I'm just looking at them right now. And the goal of every environment analysis algorithm is to now get at these niche matrices and try and extrapolate information from them and see what they're telling us about the data set that we're working with. And you could think of a few ways that you would maybe go around and try to recover some signal from them. So maybe one thing you could do is simply average these niche matrices, right? So like in Python, this is just doing dot mean and doing the gene expression average of each one of these niche matrices, and you get a simple length of gene vector that gives you some sort of representation of what went on in this environment, right? And this is something that has been done before. There's a bunch of methods that do this. I argue that maybe we've put a lot of effort into making a lot of these spatial technologies single cell resolution. And when you just do this niche compression, you're kind of losing that single cell resolution. So intuitively, you're losing a lot of information when you just do this dot mean. I also argue that even in this like toy example, we're seeing that the environment of a cell itself can be very heterogeneous, comprising of populations from a lot of different cell types. So when you average, what exactly are you getting? So we've been put a lot of effort to try and move beyond just averaging, right? And one of the things that we see a lot is people do these days is assign each cell in their spatial data set a cell type. So you say, oh, this is cell type 1, cell type 2, so on and so forth. And then your representation of the environment is just the cell type abundance in the environments of each cell in your data set, right? So In this one, you have a lot of one cell type over another, and in another cell, this environment is dominated by a different cell type. And that again gives you some idea of what its environment looked like. But when you go ahead and do something like this, which again has been done before, you're introducing bias from the word go, right? Like you have to select a resolution of which cell type, which immediately says what type of analysis you're going to do. You could have errors in cell typing, even this quote unquote very single cell gene expression example is not super solved yet. How exactly do we cell type? So you could introduce those errors downstream into your spatial analysis. And finally, on that point, I argue that a lot of these methods that went through this route of environment through cell typing, they mostly worked on spatial technologies of a couple years ago, where you could only do 30, 40 markers in a single panel. And when you can only do 30 and 40 markers, maybe the most information you could get out of your panel is cell type, right? And this is examples from like CyTOF in the single cell field 10 years ago. But now when we have spatial technologies that could easily get to several hundreds of genes in a single panel, the information that we're trying to decipher could be a bit more nascent than just cell types. So we would like to translate that also into our spatial analysis as we go forward. And to this end, what we've decided to do instead is not average and not calculate cell type abundance, but rather compute the gene-gene covariance of these niche matrices. And that gives us what we call the covert representation of the environment. And this is a step beyond just averaging, because we're not looking at like what are the genes that are expressed in the environments, but rather which genes are co-expressed in similar cells in the environments of our cell. And that gives us both an idea of cell type, and if I show you a big matrix, it's very easy to pick up what are the cell types that exist there, and continuous information beyond that. And using these covert matrices, we could construct a distance matrix that is based on the optimal transport distance between covariance matrices that gives us a continuous and principled notion of the amorphous or previously amorphous idea of environments. Now we imbue them with a quantitative representation that we can work with it. Just a quick note on this approximation. We use this approximation of the optimal transport distance to just enable our method to be a lot more scalable and to really adapt to spatial data sets that could have hundreds of or even millions of cells, which is imperative as we collect larger and larger atlases. And despite this approximation being blazing fast, and you could see here that as sample size grows, that our metric does not incur a time span of more than 10, maybe 20 seconds, even on super, super large data sets. It's also commensurate and practically has a really, really good overlap with the true Fréchet or optimal transport distance between these covariance matrices. So what you can now do is you could take your covariance matrices and you could take this distance metric that we imbued on top of them, and you could simply run almost any analysis that you would do with single cell data, but just using environment instead of expression. So you could do covariance clusters and covariance UMAP, where two cells are close together, not if their gene expression is similar, but rather if their environment is similar. So it gives a very intuitive and straightforward analysis approach to those already familiar with single cell data sets. You could also look at continuous access, like diffusion components and force-directed layouts, which is really exciting approach right now in spatial data or environment analysis, because we're now trying to get at these more continuous and nascent trends than just simply cell typing. So that brings us to the point of how can we use this representation to integrate onto single cell RNA sequencing data and imbue it with some information that didn't have before, right? So the way a lot of spatial assays are collected today is that you do the spatial assays, usually send adjacent tissues to single cell RNA sequencing, because it's just so easy to do single cell RNA sequencing that if you're already doing spatial, which is a ton of work, you might as well do that as well. So usually you have both sides of this, both sides of your assay, both spatial and single cell. Another question is, what do you do with it? And I argue that each one gives you a layer of information that the other one doesn't. So with spatial data set, each cell exists in its spatial context, which is a ton of information that we would like to harness. But you sacrifice where you can't do full transcriptome. You can only do maybe a couple hundred genes in a single slide. In single cell, this trade-off is flipped, and you lose spatial context, so you don't know where your cells came from, but you gain that you have full transcriptome information, right? So the idea is, if we have these two correlated data sets because they come from adjacent tissues, maybe we'll be able to integrate them and harmonize them into a complete data set. We have both full transcriptome information and spatial context for each cell in our cohort. So what we would like to do is impute the full transcriptome onto the partial spatial data set, and maybe more uniquely to ENVI specifically is infer the spatial context onto single cell RNA sequencing, which you had to lose to disassociate your tissue. So how are we thinking about this dual step of both imputing gene expression and inferring spatial context? Is that in the spatial data, you sort of have both, right? You have your gene expression and you have your spatial context, and you can imagine how each cell now has both this X and this sigma. The sigma is, again, this covered representation that we described earlier. And now you can learn a very simple regression problem, or regression solution, I should say, that takes you from gene expression to environment, right? Each cell has both. You have its gene expression, you have its spatial context. Just learn simple regression coefficients that takes you from one to another. Very simple. You could do it in 2 seconds with sklearn. On the single cell data, you could take your data set and subset it just to the genes that are included in your spatial assay. And now you could do this second regression step, where you take your partial gene expression and you use that to predict the full gene expression, again, learning some regression coefficients that allow you to do that. And again, this is not that conceptually challenging. Each cell has both its part of the transcriptome and its full gene expression. So you could just do X and Y, sklearn it, and you get regression coefficients. Again, 2 seconds in Python. So now you have these two sets of regression coefficients. Maybe you'll be able to crisscross them and use the regression that imputes full transcriptome and apply it onto your spatial data to see what the rest of the transcriptome should look like using the single cell data as supervision. And the flip side of it is you could use the regression that learns spatial context from your spatial transcriptomics data and use that to infer the environment of your single cell data pre-disassociation. Seems straightforward enough, I would think, right? But the issue that you run into is that if you look at spatial data and single cell transcriptomics, is that there's a big batch effect that occurs between them. They rely on different methods, seqfish and related algorithms. They use microscopy, single cell doesn't. So when you look at any particular gene and you just plot its histogram of expression, you see that they're wildly different between your single cell and your spatial data. So while we ideally would like to learn one model on one data set and another model on another data set, the distribution shifts between them prevent us from applying them on the other one. As anyone who learned a little bit of machine learning, you can't learn a model assuming one distribution and expect it to work on a completely different distribution. So maybe what we need to do instead is take our single cell and our spatial data set and first integrate them into a combined space where we don't see this batch effect, and then we'll be able to do this learning process where we learn how to impute the full transcriptome from the single cell data and learn how to infer spatial context from the spatial data. And now that we're going to do it from this some latent embedding where there's no batch effects, and this process can be done reliably. In reality, how we do it in ENVI is that we do this process fully end-to-end using deep learning and specifically variational autoencoders. We do a trick that is, it's called a conditional variational autoencoder. They do it in SCVI. They also do it in SC-Artris. I think it's very cool and very, very easy to do, where you add what's called a conditional or confounder neuron to the input layers of both your encoder and your decoder. And that confounder neuron is supposed to absorb technical shifts between any sort of two data set you're trying to integrate. And from there, you should be able to learn a latent that has no batch effect, because that batch effect was, again, absorbed by this additional neuron. So the way the architecture of ENVI works is that you have an encoder that takes as input either single cell or spatial transcriptomics expression profile, again with that confounder neuron where it has one value for the single cell data and it has a different value for the spatial data, and that encodes into a latent. That latent is then passed into two different decoders, which is one of the unique features of ENVI. One decoder is there to decode back into gene expression space. So for the single cell data, that also includes learning the genes that were not imaged in your spatial data, and that provides that imputation process we were talking about. And on the spatial data, you also take this embedding and pass it through an environmental spatial context decoder, which is trained to reconstruct the covered representation that we've been describing earlier. And after you finish training your model, what you can do is take a spatial sample, run it through the encoder and get an embedding, and then pass it through the expression decoder and look at the neurons, i.e. the genes, which were not included in your data. And that provides you with that information regarding the imputed genes, or those genes which were not included in the original sample. On the flip side, you could take your single cell data, pass it through the encoder, get a decoding, and then pass that encoding through the environment decoder and get a sort of prediction about what should have been the covered representation of that cell based on its gene expression. And this does both those two regression problems that we were talking about earlier of learning to impute full transcriptome and learning to reconstruct spatial context, and simultaneously learns a latent embedding, which is supposed to be removed of any batch effect between your single cell and your spatial data. This is a brief overview of the method. There's a few more caveats that I just didn't get into, like using negative binomial instead of Poisson, and some details with that. But both for simplicity and for time, this is, I think, a very simple conceptual overview of what it is we're trying to do. And I'll move to some results that we showed in the manuscript for ENVI. So one of the first data sets we applied it on is the Merfish Motor Cortex Atlas that was published a couple of years ago by the BICCN, where they collected 64 different slices of Merfish from the motor cortex. This atlas, if I remember correctly, had around 300,000 cells. So we're really pushing the limit of what most integration algorithms can handle because of this size. And it also had a complementary single cell RNA sequencing assay, again, of that same tissue. So it really provided a nice case study for us to apply our algorithm over. So first thing that we want to do is you want to take our two data sets and integrate them into a combined latent. And then it's very nice, we could just plot that latent and see what it looks like and make sure that similar cell types co-embed in that latent. And we find that they do. We can see the excitatory neurons in the two data sets co-embedding, also by their cortical layer, interneurons, non-neuronal populations, even ones that are super, super rare, like these oligodendrocyte progenitor cells, even though there's maybe only a couple hundred of them in the single cell data, they integrate rather well with the Merfish data. But beyond that latency, we'll like to see, okay, tell me something I didn't know. Don't just integrate the data, provide me with something new. So first we want to talk about the imputation, which I think is a very standard thing that people want to do when they have spatial data. So what does imputation look like is that you have your image genes and you use the single cell data to supervise and give you gene expression of features that were not included in your assay. We generally want to say how good did we do this, but we're kind of struggling because there's no ground truth to these unimaged genes. So what we can do instead is take our spatial data and sort of artificially divide it and take a few of the genes and hide them from our model and then impute their prediction and see how well we did compared to the ground truth expression of those genes. And there's a few algorithms that do it. We find that ENVI does better than all of them. I think, to really answer how well we did, someone who isn't me should do a more comprehensive benchmarking study because I'm obviously biased to what I did. But we tested this a bunch of times. It looks like we're doing pretty well compared to the ground truth. And even when there is no ground truth and a few of these genes were not included in and these genes, for example, were not included in a data set. But when we look at the Allen Atlas, which did individual ish imaging of those genes, it looks remarkably close to where they're supposed to be. So DCLK3, we predict it to be in the shallow layers of the cortex, and that's what it looks like to be, so on and so forth. I think the flip side of what we're trying to do, which is infer spatial context, is a bit more nuanced and also something that hasn't really been tackled before. There's a few algorithms out there, like these guys, that get at gene imputation. We're saying we can do that, looks not that terrible. What can we do beyond it? So here's a nice little vignette from it where we take the somatostatin interneurons, which are a group of inhibitory neurons in the cortex that are very, very critical to the neural circuitry in the cortex. And when we look at those that population in the single cell data, because it's full transcriptome, we can actually label them further into specific subgroups within those somatostatin interneurons. Those names are just based on marker genes that we see those populations express. But in the spatial data, those labels don't exist simply because there isn't enough gene breadth to assign these interneurons any further labels beyond just saying they're somatostatin positive. So we know that this population exists. We know that it's very, very heterogeneous, but we can't say where it came from because that doesn't exist in the spatial data. But what can we do instead is we can take the covered predictions of the somatostatin interneurons and integrate them with the true covered representations of our spatial data and see what that tells us. What we find is that the first diffusion component and the primary axis of the force-directed layout of the somatostatin interneurons remarkably corresponds to the cortical depth of those neurons, which allows us to assign each cell in the single cell data a sort of predicted pseudo depth. And when we look at how that groups within the specific subtypes of these somatostatin interneurons, that sort of gives us a prediction of where we think those somatostatin interneurons came from. So we say that CALP2 positive somatostatin interneurons came from shallow layers of the cortex, whilst TH positive interneurons came from a lot more deep inside it. And it was kind of remarkable because while we were working on this during the revision of ENVI, this paper by Gord Fischel from Harvard actually went ahead and imaged some of these subtypes using genetic mice models. And what they find corresponds perfectly to the ENVI predictions. So they see, using their models, that CALP2 positive somatostatin interneurons are indeed localized to shallow layers of the cortex, and CRH positive SST neurons are localized deeper inside the cortex, which matches perfectly what we say. And because we have full transcriptome single cell atlas, we can actually provide a bit more predictions than what they could do with molecular imaging. And these serve as like further hypotheses that we believe could be validated with other experimentalists later on. Now I'll go very quickly into a few Xenium results. Xenium, for those who don't know, is new commercial spatial technology by 10x. This was, I think the data I'm showing here is the first panel that we collected in the Pear lab of Xenium. So we're only doing 240 genes. I think now they could do a couple hundred more, but we went ahead and did Xenium and single nuclei RNA sequencing of a leptomeningeal metastasis to the brain, melanoma metastasis to the leptomeninges in the brain, which is both an interesting new technology that we want to see how good a method does on, and also it's a pathological case because this is a metastasis. There's a tumor in the mix, which throws a whole wrench into how we're thinking about this data. So we have this data set, we integrate it, and we find that despite this pathological context, most cell types co-embed remarkably well, including the tumor population, which is a complete pathology. It's not something that we designed our model explicitly to take into account, but even despite that, it does so rather well. So we do the integration pretty well, but there's kind of a lie in this image because when we look at the immune cells and the single cell data, similar to what I showed in the previous example, we find that they're a lot more heterogeneous than what could be deciphered by the spatial data alone. for the immune cells, specifically in the context of metastasis to the brain, are comprised of two distinct groups. There's the brain-resident microglia and the tumor-associated macrophages. Now, in the single-cell data, because we have full transcriptome, they're very well delineated. We can assign labels to which cells of the immune population are microglia and which are macrophages. But in the xenium, simply because specific myeloid markers were not included in the assay, it's it's impossible to tell which is microglia and which is macrophages. So we sort of have this problem where we know these populations exist, but the spatial data alone does not provide us with enough information to say where they come from. But when we look at the covered predictions, we find that there's three main environment clusters. One are immune cells that are localized to the cortex. Another is immune cells that are localized to the basal ganglia. Another one is immune cells that are localized to the tumor region. And when we overlay the single cell labels onto these COVID predictions, we find that the macrophages are predicted to be precisely in this tumor-associated cluster, which is really what we're expecting to see. It's like these macrophages are associated with the tumor, and we know that they localize to the boundary and inside the tumor. And we can use cell type to sort of guide what we expect those niches to look like. And the macrophages indeed have predicted to have mostly tumors in their environments, while microglia are predicted to be more dispersed in the brain, mainly around the basal ganglia and the cortex. So this is a brief overview of ENVI and COVID. So there's the one algorithm, which is just for spatial data, which is to represent spatial context. The next step is we take COVID and imbue that onto single cell data with ENVI, which allows us to map discrete and continuous spatial signals onto single cell RNA sequencing data. Because this is SCVerse, I just want to say a brief remark on the code. I wrote it in JAX, which was a lot of fun because the documentation for it is kind of hard to get through, but that allows it to be really, really, really fast, like training it even on the massive multi-cortex atlas with hundreds of thousands of cells took only around 4 minutes. It works with ScanPy and data, and we try to make it as easy as possible. So you only have to give the code just your two single cell and spatial and data, and it does everything else for you. So you could just read out the results without having to think about it too much. I want to thank everyone here for inviting me and for coming to this talk, and a big, big thanks to my lab and collaborator labs that allowed us to do this study. Thank you.
Thank you so much for the great presentation. Team, you're actually clapping. I will not comment on the lack of default parameters, and nowadays it's a hot topic, so I'm gonna leave it pass. It's good that you tune the default parameters, but let's not go there.
Let's not get into. Let's not.
I agree. Also, it's recorded. The presentation is gonna be on YouTube. So I will cut this part. So. I will invite first Tim because I saw also he raised his hand to ask his question. And then I will move
on. Yeah, one of the questions was like just very, very in the beginning where you showed kind of like the performance thing, where you had like the time versus samples. In this case, you're calculating one niche per cell.
Yes, one niche per cell.
Okay, so this was like power 5 niches.
That's a great question that we keep getting over and over again. It's not well defined. I'll just say this. What we did in our work is we took every cell and we just take its 8 nearest neighbors. And that's what we call the niche of a cell. But that is obviously, speaking of parameter fine tuning, that is a parameter that maybe you should fine tune specific interest, right? When you look at, these small numbers of 5, 8 nearest neighbors, you're mostly looking at like the sort of signals you're trying to get at are, at least how we thought about it, was cell-cell interactions. But, say you're looking for broader architectures in your tissue, maybe you want to go to even the hundreds of cells in your environment, right? And try and see what that means. As briefly as I can say this, it really depends what you're trying to get at. For us, looking at 8, and we found that the results are pretty robust around that number. Like if you choose 6 up to 16, it all looks kind of the same. But say you want to find some larger patterns, maybe you should go much, much higher than that.
Okay. Yeah, I guess I was just trying to get like a rough feeling about like, is it the cell type number or is it just the niche? Because like you can have 100 million cells if you discretize them to like 100 niches. The runtime is the same, right?
So the runtime is constant. Yeah. Like even if you have 10 million cells, okay, like
But the number of niches is the important thing, right?
So each
Like
cell has one niche. Yeah. Like each
Okay.
cell, we just take its nearest neighbors. You could think maybe you want to get at like some multi-scale approach where you look at like multiple different environment or niche views for the same cell, but we didn't get into that in our work.
Okay, thank you.
No problem. I'm reading one of the questions in the text is Alex Lung asked, can this only work if single-cell RNA-seq experiment and spatial experiments are complementary? Could we infer impute data if I only have spatial experiment and we use this with human atlas data? Great question. So we looked into it a little bit when the data sets are not exactly complementary. And by not exactly, I mean like they're not, say, adjacent tissues or even collected by the same lab, but they're usually representative of the same context. And it looks like it works then. I'm not sure how it will work if you have like one data set that is like this, one data set that came from one study and a completely different atlas, how well be able to integrate. I would maybe think that you could take that atlas and subsample it just to these cell types that you expect to see in your spatial panel, and maybe that could be used to integrate the two data sets. Good question. I'm not really sure yet, but we do know that it doesn't have to be like a perfect one-to-one mapping for it to still work rather well.
Good. We skipped one. I'm going through the chat. So there is one more. Could you please elaborate a bit on how the imputation correlation was calculated? Okay,
great question. I think it's very, very interesting to ask how similar are two expression profiles in spatial data, right? So one way you could do it is you have all your cells and you have the gene expression predicted by ENVI and the, say, ground truth gene expression, and you could just say compute standard Pearson correlation, and that gives you some information of how close these two gene expression profiles are. We did that, looks good. We also took a bit of inspiration from computer vision. There's this algorithm called multi-scale structural similarity measure. I can type it. It's called MSSSIM, something like that. And what we did as well is sort of like look at gene expression in multiple different scales of our data, and we compared how it looks as average on those different scales, and that gives you like another score of similarity, where the goal here was to take into account like the spatial structure of our spatial data when we compare gene similarity. Both of these metrics showed similar results, that ENVI does better than some other algorithms. Like I said, I think, I can't judge myself. I want my method to work well, and I want other methods to work not as well. So, take my results with a grain of salt. But I hope that when a comprehensive benchmarking study comes out, they'll use a few different methods to look at how similar to gene expression profiles. And I really, really hope that ENVI does well in that case as well.
Thank you very much for your presentation. It was my question. And yeah, like I've been testing ENVI for the past few weeks. And as you said, we did some hidden genes tests. And I also tried MSSIM, but
yes,
for us, it was like huge problem with the RAM memory because it took more than 40 gigabytes of RAM or something like this.
I think I know why. I should have added that in the tutorial. You need to like add a batch key, because else it would like think that all the data came from like one sample, and it'll try to compute like a massive graph based on that. But what it's supposed to do is, I assume it took so much memory because you have a really big atlas with a ton of samples, is do this like individually for each sample.
For each sample or for each cell type?
Each sample. Like,
Each sample.
but it, because like, one sample, you know, your environment is only within that sample, right? So you just, it's a hyperparameter that I need to add there. I'm sorry it didn't work. It should work if you add that parameter. Email me later and I'll go through how it should, like how it's supposed to be run. But I didn't tell people that.
Yeah, no, I totally understand. I mean, we also tried Spearman correlation and it wasn't also like amazing. And I assume it's because we run on hard tissue, and as I understood, you only did mouse brain tissue, right?
We did a few. We did mice brain, and we also did a gastrulation, which is a whole mice embryo.
Okay. So do you think that the thing that mouse brain is super like domain based might affect the analysis of other tissue like heart, which is very heterogeneous and not so like you cannot see domains purely?
Yeah, I hope not. The reason there's so much brain in our paper is because that's what most spatial data sets out there are.
Yeah.
Like it's the easiest tissue to image.
Yeah.
So like if you look at each time a new technology comes out, it's always brain, it's always cortex, it's always something like that. So that's why we had a lot of it. We tried to include like the first two figures in the paper are from the Marioni's gastrulation sequence, which is not brain and is maybe more closer to what you were thinking of, where it's not like as delineated into layers like the cortex. I hope it works well. We need to test it some more on newer data sets as they come out. That's the major thing.
Please publish it somewhere. It would be super interesting.
Yeah.
If I may, a few more questions, if I'm not.
Go for it.
Okay, cool. Yeah, so what I also saw in the data is that even if in single cell original atlas, some of the gene expression is very low and in like less than 10% of the cell in this particular cell type, after we run ENVI and impute genes, the expression is still low, but like in almost like 80% of cells in this particular cell type. Is it like, am I doing something wrong, or it's just the thing that did it happen to you as well? Like, can you say something about it?
This is like, this like connects to like, how do you pre-process stuff best for this? When we ran this, we tried to make it like as broad as possible. And we took the data sets so like the Xenium data, the only which was the data set that we collected ourselves. The only pre-processing that we did was, it's been a minute, but was removing mitochondrial genes, library size normalization, and filtering out doublets. So there wasn't like, you know, magic imputation or any algorithms that go into that. And all other data sets, we just took the data as is. We downloaded them from the repos and ran the algorithm without any further pre-processing. Maybe for a couple of them, some cells were definitely bad, so we just removed them. I think there is obviously like when you do those steps, it's going to introduce some sort of bias into the algorithm. We tried our best to not introduce any bias beyond what was the intent of the original authors. And it looked like it worked okay. But imputation onto the spatial data definitely depends a lot on the quality of that gene in the single cell.
So do you
this is a
less, oh, sorry.
Ask, it's your question.
Like, do you suggest as less filtering and pre-processing as possible, or?
Like I said, we did that because as far as when we did this, we took the data as they were provided to us and didn't want to do any processing beyond that, because then it's like, it's sort of like cheating as far as we're concerned. But when you run this yourself, you should process your data as you see fit to get like the cleanest, best representation that you can think of, and then that should be able to integrate with ENVI.
Yeah. But at the same time, both single cell RNA and spatial counts should be pro, right? So you would don't normalize.
Yes. So ENVI does not assume that the data is not logged.
Mm-hmm.
But you could process it and then just like take the your dot counts obsam and use that instead. It shouldn't affect your processing. Something that we didn't elaborate on, but I think is interesting, when we look at results, say from other gene imputation algorithms and compare it to ENVI, if you say plot like the score of one versus the score of another, I'm not sure which axis especially, I don't know if it's merit, but if we do score of one versus score of another, it's very well correlated.
Mm-hmm.
Like maybe, like broadly we see that ENVI does better, but if a score of ENVI is high, we also see that the score of like Harmony or Tangram is also high and vice versa. So those are like, it says that your ability to impute specific genes is very related to both the method you're using, because some methods do better, but also some intrinsic factors of that gene that makes it less or more imputable, quote unquote.
Okay, last one and I'm out. Do you say, is there like a minimum amount of counts that should be in your data to be imputed correctly? Because like in mouse brain, the amount, like number of counts is definitely higher than in heart or like kidney tissue.
Yeah. I don't know, like I can't give you a specific number, like how many counts you should expect. In one of the data sets we used in the paper, which was brain, but it was like Stan Anderson's awesome fish data from like, at this point, like close to a decade, I think, which was very, very small, like only a couple thousand cells, only 30 genes, something like that, and was super sparse as well. And it still looks like it worked well. The one caveat is that we used a different modeling distribution. We had to like use negative binomial for the spatial instead of Poisson to account for those that inflation of zeros, but that seemed to fix it.
Yeah. Okay. Thank you very much.
Thank you.
I want to comment really fast that I have been working with liver tissue and spatial data. I really believe that the reason they use brain for the technology is because it really looks good, and all the others, they look not very good.
Yeah.
I don't believe that just happened.
I'll tell you, on that note, one of the issues we've been having a lot when we're trying to run Xenium now is segmentation. And like brain is much easier to segment because it's a lot sparser. But when you try to segment any other tissue that is dense of nuclei, it's like it's a whole mess.
Very diverse also in the cell types, and while the others they look more similar. Anyway, so 2 more questions, 3 I see. You have to be fast so we can do it in 10 minutes.
I'll go for it.
So 1, why is it important to infer the genes not in your panel? I thought the 1K plus plex panels that are starting to get available, could you not just work on the intersection of your single cell assay and spatial assay genes? It's also on the chat.
Why is it important to infer the gene? Okay, so I completely agree with that point. That is, I didn't want to, I say it like in-person talks, I think the gene imputation problem is a bit outdated. Like we show a bunch of them in the paper. Like I show like DCLK3 and Cortex. But like if you're super interested in what does the expression of DCLK3 look like, you just include it in your panel. You won't go through this contrived way of like, oh, let's collect single cell and run all these methods to see what it looks like. We just add it to your panel. I agree completely. I think it's cool to do. I think it's what people expect to see. I also think it's not hard. Like when we went and built this, it wasn't like a conceptual leap to just include that feature into the algorithm. So we were like, great, people are gonna love that and use it because people know how to work with gene expression. I do think that we're at the point where you could calculate image almost any gene you're interested about. So you should just go ahead and do that. But the conceptual novelty of ENVI was the other side of inferring spatial context onto already full transcriptome, single cell data, which is something that other methods, I think, sort of overlooked up until now.
Good. Next one. How big of a focus does ENVI put on selling the imputation aspect? Imputation is a hot topic here.
Yeah.
There are quite a few more methods which you could compare against that. Also use VI and perform reasonably well, like Mephisto.
Could you repeat that regarding imputation?
Yes, so it's also on the Q&A, if you can see it. How big of a focus does ENVI put on selling the imputation aspect?
I think I must not be seeing that message.
Wait, I can put it on the, wait. Do you see it?
Okay, yes. So yes, there's a lot of methods that do that. We benchmarked against, I think, in total like 8, which was, you know, a bit of work on our behalf because there's a lot of data sets and a lot of other methods. Yeah, there's a lot of other methods that we could compare against. I said, like, I think that I'm not the best judge here because I want my method to work best, obviously. So like, we didn't do any sort of like handling or stuff like that, but maybe some of the other methods could have been run a bit more optimally for the different data sets that we used on. I'm not entirely sure. I think that there's other groups right now that are more working on comprehensive benchmarking essays, where that is like the entire goal of taking, you know, not just the 8 methods we did at ENVI, but all the 20, 30 methods that do imputation and running them as you're supposed to run them and not just how I thought I need to run them and giving a precise result and like what does better, what does worse. Regarding how big of a focus do I put on selling the imputation, I don't. I think it's outdated. I think there's cooler problems to try and solve right now.
Good. And the last one, do you plan on sharing your code you used to get the correlation results in the paper?
Sure. I mean, the ENVI code is out, but the specific it's notebooks that I use to just run this. I can share it. If you shoot me an email, I'll send them to you.
Good. So many questions. It was a very interesting one, but
thank you.
Yeah, thank you so much for giving this amazing talk, and thank you everyone. I have shared all of your information, and I think everyone can find you, like Twitter. And are you only Zulip? I think you're on Zulip as well, right?
I'll add myself to it.
Okay, good. You should have said yes and just do it, you know.
Yes, I'm on Zulip. Because I like looked for the link and I clicked on. I was like, okay, I need to join this.
Good. So now he's on Zulip, and I shared GitHub, and feel free to reach him, and I'm pretty sure he's more than happy to reply on everything. Thanks again for joining, and see you in 2 weeks for the next community meeting.
Thank you so much. Goodbye, everyone.
Thank you very much. Bye-bye.